Hey there friends, I am Lily and welcome to Ecology, where we break down the science of nature. Today, we are diving into the watery world of keeping ecosystems balanced, fishes feeling healthy, and mermaids staying fancy. Strap your goggles and let's explore the physical and chemical properties of a freshwater system. Different physical and chemical parameters are used to assess the health and current condition of a freshwater body and the organisms living in it. Before we begin with the assessment, let us get familiar first with the instruments to be used in assessing the health of a freshwater body. For a little brain teaser, can you name the instruments presented on the screen? The instruments flashed on the screen are used to determine different physical parameters in a freshwater body. We will go by them one by one for us to get familiar. For the chemical parameters, let us see if you can name them. The instruments flashed are a combination of instruments and a test kit will be utilized. We will go by them one by one for us to get familiar. Now that we have seen the instruments, shall we put science into action? Beside me is a pond where tens of my friends can be found. We shall start by assessing the physical parameters of their home. Come and swim with me in this adventure. First, we shall determine the color and depth of the pond using a Secchi disc. The Secchi disc is a circular-shaped disc usually 8 inches in diameter and has 4 quadrants containing alternating black and white colors. To determine the depth, the instrument is lowered to the water until it becomes invisible to the human eye and lifts the disc enough for you to spot it. The depth between the disc is seen and not is called the Secchi depth. Now, we can determine the color of water by determining the color of white quadrants of the disc and match it with the closest color found in the scale. For today, we will be utilizing the scale provided by Forel Ule. Light availability affects different ecological processes. They are important to understanding freshwater ecosystems. In measuring the light intensity, a light meter is used. Turn on the light meter and expose the instruments to the source of light. Then wait for 1 minute and record the reading. Perform 3 trials to ensure accuracy of the data and calculate the average value. Light intensity is crucial as it affects the rate of photosynthesis and distribution of different aquatic plants. The rate of light intensity varies depending on the depth of the pond, and the ideal light intensity ranges from 200 to 500 lux, allowing healthy plant growth without causing algal blooms and still providing enough light for aquatic organisms to thrive. Water temperature affects the health and productivity of aquatic ecosystems. Let us measure the water temperature using a pen-type thermometer by submerging the probe in the water and waiting for it to stabilize for about 1 minute. Remove the instrument from the water body, record the result, and repeat the process for 3 times to ensure accuracy. Measuring the temperature of water is important as it affects the growth and development of different aquatic organisms, thus altering the distribution of species, the availability of oxygen, and the dissolution of hydrogen ions. Generally, the optimum water temperature ranges from 20 to 25 degrees Celsius. Now that we have been acquainted with different instruments and physical parameters in a freshwater body, let us begin to dive deep into the different chemical parameters. Let us determine the pH of water using a pen-type pH meter. To use this instrument, submerge the probe into the water for 1 minute, wait for the reading to stabilize, then record. Repeat the process 3 times and get the average. pH greatly affects freshwater bodies due to its ability to change the state of pollutants and nutrients to be available in water, affecting the aquatic species exposure. to optimal pH ranges from 6.5 to 8.5, allowing the species to thrive in the environment. Lastly, the pH of a water body is affected by temperature, where higher temperature would decrease the water's pH, making it more acidic. Fresh water naturally contains salt in a very low concentration. We measure the amount of salt present using a refractometer. To use the refractometer, first, collect a small amount of sample from the freshwater body, open the prism, then add a few drops and close the cover. Position the instrument facing a light source, look through the eyepiece and locate the boundary where the line crosses the scale. The salt content of freshwater bodies should range from 0 to 0.5 parts per thousand. A very low salt concentration ensures a healthy freshwater ecosystem. Dissolved oxygen is the amount of oxygen present in a water body. Aquatic organisms consume oxygen in its dissolved form. Thus, it is crucial to measure this parameter in order to assess the potential of a water body to accommodate different life forms. In measuring the available dissolved oxygen in a water body, a DO meter is used where the probe is submerged into the water for 1 minute, wait for the reading to stabilize, then record the reading. Repeat the process for 3 times and get the average reading. In a freshwater body, the optimum range of DO is around 8 milligrams per liter to ensure that aquatic organisms thrive in the water body. Let us now proceed in measuring the different nutrients including ammonia, nitrate, and nitrite, which is measured using an API kit. The instructions, instruments, and different chemicals are provided for us to determine the condition of a freshwater body. The 3 nutrients measured were ammonia, nitrate, and nitrite. Ammonia came from food residues and organic matter. The value of ammonia should not exceed 0.25 parts per million. Nitrite comes from the decomposition of organic matter by nitrifying bacteria called nitrosomonas and should not exceed 0 parts per million. Lastly, nitrite is converted into nitrate by another nitrifying bacteria called nitrobacter and should not exceed above 50 parts per million. These nutrients are part of the nitrogen cycle, nitrate being consumed by aquatic plants for their growth and survival, while ammonia is the product of these aquatic plants and is soon converted into nitrite. In freshwater bodies, these nutrients should be maintained in low amounts to ensure a healthy environment for aquatic species, supporting their growth and survival. And that is it, friends. You are now equipped and knowledgeable enough to assess the physical and chemical properties of a freshwater body. See you until the next fieldwork video.
She knows a lot about fieldwork stuff. Lily's here to explain. She knows a lot about fieldwork stuff. Lily's here to explain.